A nossa convidada é professora no Departamento de Bioprocessos e Biotecnologia, na Faculdade de Ciências Farmacêuticas da Universidade Estadual Paulista. Valéria, muito obrigada por ter aceito o convite e por estar com a gente aqui hoje. Certeza vai ser muito interessante a tua palestra. So, thank you, Valéria. You can start now.
So I'd like to say thank you for Ana Paula and Maria for the invitation to make this presentation for you about a little bit about what we're doing here in Brazil. So today I'm going to talk a little bit about Bioprocess, microbial cell factory. And so to introduce, I'm going to say what is Bioprocess. So you can say that it is a specific process that uses completely living cells or their components, for example, bacteria, enzymes, chloroplasts, to generate value-added products. So what do? You give raw material for the microorganism, and they made a lot of metabolic roads to make our product. And in a very simple way, you can say that if you are working with aerobic microorganisms, you are going to give to them oxygen, a nitrogen source, carbon and energy source, minority compounds. And so the microorganism will metabolize these compounds, make different biochemistry reactions, and as a result, we are going to have carbon dioxide, biomass, and biocompounds. And if you think about which is these biocompounds, you can say that you have a lot of products that we are using today. For example, antioxidants, growth hormones, enzymes, bioremediation, fermented food, organic acids, colorants, herbicides, alcohol, fungicides, insecticides, antibiotics, anti-cholesterol, other types of medicines and food additives. So we have several compounds that you are using today that are produced by microorganisms. However, you can also improve this production. And how we can improve it? You have the chemical diversity of microorganisms that can be used to select producers that will make our product without too many bioproducts that can be not so good for the downstream process and without toxic products. This second point is very important when you are working with bioproducts that's going to be applied in pharmaceutical stuffs and at food. And you have several kinds of cultural collection around the world that have different kinds of microorganisms that are able to produce these compounds with less bioproducts and without toxic bioproducts. And today we can use the taxonomy to see how microorganism is more similar to one that we're using in order to select the best one. If you think about a process in general, you can say that you can share it in 3 phases. The first one is the selection, when the microorganism is selected. The second, the cultivation, when you have determination of classic parameters. So how is produced our biocompound? How is the growth of microorganism? Which is the best carbon and nitrogen source. And after this first screening about the classic parameters, you have to make identification of new products or product pathway. We can say that this identification of new product is not so easy. For example, I am working with biomedical since my PhD many years ago, and until now we don't have the definition of the chemistry. I hope that this is going to be made this year in collaboration with Sonia from PATH. However, these kinds of identification, mainly when you have biocompounds from fermented broth, is not so easy. However, it's important because after you can define the best route, metabolic route through the product. But this is the first screening that you have when you are working with a new product. And the cell factory, when you are working with microbial cell factory, which is the characteristic. These cells, they have to metabolize a different kind of substrate, and they have to have a good metabolization of the substrate. So we're not going to have a lot of substrate in the end of the fermentation, and it is going to use this substrate to grow and to produce our target biomolecule. It's interesting if high productivity, if applicable for bioreactors. So if you are working with some products that are going to be used industrially, you are going to have it producing a high amount. So it's not so good work only incubator shaker. The best way is working bioreactor. However, bioreactor is a new step that sometimes you can have different problems that I'm going to talk a little bit after. And it's good if this microorganism can be changed for genetic engineering to the gene pass for different hosts that the metabolic road is more known. So after the first screening, they have to begin the second screening. So now you have the microorganism and you have to begin the new phases. So incubation, incubator shaker, incubation in bioreactor. So incubation in incubator shaker, we can say that is small scale, while bioreactor is a scale up and you're going to have a high amount of product produced. When you think about experiment in incubator shaker, it's carried out during shake flakes. So what's good? It's good for make a medium composition. So what is the best carbon source? What's the best nitrogen source? What is the minority compounds interesting to be included in the medium fermentation? Because we can do several experiments in a very short time. And in this point is that you also determine the initial pH of your cultivation. So you are not going to control the pH through the fermentation process. However, with the best pH in beginning of the fermentation. We know that during fermentation, the several microorganisms can produce organic acids that's going to make the medium more acid, or ammonia, for example, as subproduct that's going to make the media more basic. And so we're not going to control, but we are going to know how is the initial pH, which is the best temperature for the microorganism grow, and therefore the microorganism produce our target biomolecule as well. And you can also define other environmental conditions allow optimal productivity. In around these experiments in small scale, you can calculate some parameter as cell growth rate, specific productivity, and product yield. So after the end of these experiments, we are going to know about the performance of the organism, how the organism grows, how is the production of the target biomolecule. When we pass for the second step about cultivation in bioreactor, we are going to make this scale up. And when you are working with aerobic microorganism, environment very important are agitation and aeration. And after you can transfer these and make this comparison using the volumetric oxygen transfer coefficient. That's a very important parameter when you are working bioreactor, and the time of production. In this point, define different parameters. So the first one is the kind of bioreactor we are going to use. So in this photo, I am showing for you the starry tank bioreactor. That's the most traditional bioreactor used in the production of biomolecules. However, you can use, for example, airlift bioreactor that's more suitable for filamentous fungi, for example, and we have other kinds of description of bioreactors. Besides the kinds of bioreactor that we are working, you can use here, for example, the mode of the process. For example, if you're going to use bat, feed bat, if you are going to work with a continuous process or discontinuous process. So this kind of parameters is going to be defined in this point. So it's not so easy make this transition from incubator agitator to bioreactor, because you have to select different points. So in incubator shaker, you define the media composition, you define the best temperature, and after in bioreactor, you have to define, for example, the best agitation, the best aeration for your microorganism. In bioreactor, you generally you want to make a high production of the target biomolecule in a less time and in the high production that's desired. Here you can also calculate some parameter as cell growth rate, specific productivity, and product yield. And in bioreactors, so you can say that we will improve the production of the bioproduct by microorganisms. So we have this point about the production step. In this point, we can make a steady improvement. You can work to direct the metabolism of the microorganism to higher product formation, and you have to eliminate the bioproduct formation as well. If you make this, it's going to be much more easier. This is the point that you can say it will work first in incubate shaker, second in bioreactor with a small volume, and after a bioreactor with higher volumes. But if you work very well with these steps about the production, you can improve the downstream process. So, for example, if you are working with byproduct from the agro-industry, so in Brazil it's very common to have several works that was with, for example, bagasse from sugar cane to as carbon source for the microorganism to produce other bioproducts. When you do this, what you have? The microorganisms, they have to make the metabolization of this bagasse. So they have to produce several kinds of enzymes, for example, chelanes, cellulase, to metabolize the bagasse and after use this carbon source to produce other biomolecule. As a result, you have a fermented broth with a high amount of a bioproduct. And when you go to our downstream process, we have problems because you don't have only our target biomolecule, but several biomolecules. Other way, the microorganisms, they have to make the metabolization of the product because one point is that they have to grow, and to grow they have to produce several metabolites, and these metabolites is going to be present in the fermented broth, and when we are going to make this downstream process, even they are going to be contaminants of our target product. So if you work in the production step, reducing the bioproducts of the process, we are going to have less contaminants in the upstream process. So it's very important you have the knowledge about how was the cultivation of the target biomolecule to define the experimental conditions in the downstream process. Now, today I'm not going to work to talk about the downstream process, but only to clarify each point. If you're going to take a fermented broth to make the downstream process, it's very important you know, for example, which is the media that was used to produce this specific biomolecule, which is the proteins that it can produce, which is the other contaminants that can be presented. So after this first selection, you can work and make the strategy to the downstream process. Okay. And so today I'm going to talk a little bit about one work that I have been doing since my PhD, that I use filamentous fungi to produce different kinds of biomolecules. So, and why I want someone going to say, oh, filamentous fungi is too hard to work because they need a long time to make the fermentation. So it depends on, you can use 7 days, 15 days to produce the biomolecules. But the filamentous fungi, we have several fungi in the nature, and they have physiological and biochemical diversity. And for this physiological and biochemical diversity, they produce several kinds of biomolecules. They are eukaryotic organisms, so they produce a lot of more complex molecules, and they don't need a lot of, they don't need nutritional requirements very they need low nutritional requirements. So for this, they are interesting. And we produce several kinds of products. So for example, enzymes, antibiotics, alcohol, food additives, and chemicals. Inside the chemicals, it's one that I'm going to talk for you today. That's about the colorants. That's what I'm going to working in our research group working for several years. So the colorants are molecules used to impart color, enhance or restore the appearance of products, which are used extensively in the food, cosmetic, pharmaceutical, and textile industry. If you look around us, we are going to see that the color is very close to us in several products. So not only in our food, but in our clothes, in our pharmaceutical, those stuffs that we have. And for many years, the synthetic colorants were used in the industry because they have high stability, and the main reason is what the price, because synthetic colorants are cheaper than the natural ones. However, in the last years, several synthetic colorants that was being using began to not be more allowed to the regulation organs. So in Brazil, you have Anvisa, USA, you have FDA, in Europe, you have EFSA that define which colorants and other additives can be used in the product. And these synthetic colorants began to be not so used, began to be not allowed to be used in different products. In this sense, the natural colorants began to grow, and the interest began to grow as well. So the natural colorants can be produced by plants, insect tissues, and microorganisms. Why is it interesting produced by microorganisms? Because if you can, after the determination of the parameters, we're going to have a production in a batch process with not so many variations. So the production is going to be established, it's going to be stable. Generally, the natural colorants produced by microorganisms are more stable than plants and insect tissues. However, when you think about natural colorants produced by microorganisms, you need yet and very studies, because you don't have a lot of in the industry, you don't have a lot of colorants approved by these regulatory agencies, and so you need several studies. One of the species of microorganisms more studied that produce colorants are monoscous species that are allowed to be used in Asian countries. However, is not allowed to be used by FDA, FSA, and Anvisa. And because although monoscous produce different kinds of colorants with color very high, they produce also a mycotoxin called citrinine. So they have several studies that studying this production of citrinine in order to reduce the production of citrinine. However, new species that can be used as self-factory for safe and reliable colorant production is also interesting. In this way, we have the Thalaromyces species that are growing, and the number of the production, the studies about the production of colorants by Thalaromyces. And one that I'm working since my PhD is called Thalaromyces amistokii. So Thalaromyces amistokii, they produce red colorants in plate agar. For example, this is one cellular structures of the microorganism, and this one the production of the colorants. And they are interesting because they are stable in alkaline conditions in the presence of salts, polymers. They have antimicrobial activity, and they have antioxidant activity as well. So I'm going to talk a little bit now about some work and how we improved the production of these colorants. So here is the works that I made during my PhD, and the name here is Penicillium purpurogenum. So it was the first name of the microorganism, but after a taxonomy revision, the name of the microorganism changed for Penicillium purpurogenum to Thalaromyces amistokii, but it was only a taxonomy problem. In these experiments, what we did? We did experiments in incubator shaker, and we could define the kind of inoculum that we are going to use. Is more useful use spores when you are working with filamentous fungi, but here we standardize the agar mycelial discs to produce the colorants. We defined the hours of the submerged culture and defined the carbon and nitrogen source. So in these experiments, we use sucrose as carbon source, yeast extract as nitrogen source, and after a statistical approach, we have the production of 3.59 units of absorbance of colorants. This point, when you have color, is very commonly used. We calculate the quantification of colorant produced in spectrophotometer, and we can use the absorbance of these compounds to define the amount of colorant produced. So for this, we use unit of absorbance. However, at this point, we had different kind of problems. The first one is the chemical structure of the colorants that was not defined. And we realized that although the microorganism excrete a part of the colorant produced, a high amount it still begin in the cell. So we have intracellular colorant produced, and we had low yields of red colorant productions compared with the production of other microorganisms and the production that is desired by the industry. So we began to work with different purpose, and one thing that you know is that the combination of the carbon and nitrogen source is very important, is a key factor for the production of the colorants. In the first screening that you have made, we saw that for sucrose, yes, extract was the best nitrogen source. But you begin to think, if you work with different kinds of carbon source, we are going to have other nitrogen source that can improve this production. And so it was showed in this manuscript. So we done experiments in incubator shaker, and we employed glucose as carbon source. As a result, we have improvement of the production of the colorants when you work with meat extract and meat peptone. So what you saw, when you have sucrose as carbon source, the nitrogen source is yeast extract. However, when you have glucose as carbon source, the best nitrogen source is meat extract and meat peptone in combining. So we made a statistical approach here in this point, and we could improve the colorant production. However, we had the same problems: chemical structure of red colorants, intracellular colorants, and low yield. In this point, we began to make more experiments about the chemical structure, and we could define the main chromophore, the main structure of the colorant, but not also the complete one. We have problems with this kind of we have problems with some points in the characterization, but you have the main ring of the chemical structure. So we began to think a little bit more, and we saw with the main chemical characterization that the metabolites produced by Thalamycin stokii was very similar to metabolites produced by Monesco ones. And this, what is present for you, is the metabolic route for the production of Monesco's one. So they are azathioline polycetides colorants that is produced from with the use of polycetide synthase and by a transesterification with fatty acid synthase that are all produced during the fermentation process. So which is the first colorant produced? When you saw that the first colorant production was the orange one, and after amination process, the red one is produced, and if the orange colorant is reduced, you have the yellow one. So we have this point. And so what I think, if you give a different kind of nitrogen source for our microorganism, maybe the production of the red one is going to be improved. And so what is we did in the next studies that was also published, what do you use? We would like to replace the complex nitrogen source, meat peptone and meat extract, for other nitrogen source. So we analyzed ammonium sulfate, ammonium nitrate, sodium nitrate, and monosodium glutamate. We have a very good result here using monosodium glutamate. And what is the best thing? We saw that the microorganism did not grow a lot, and the most part of the colorant was extracted, was now in the fermented media. So we not have a lot of amount of colorant intracellular. So this was a very good result. And after we made a statistical approach to analyze the variables glucose, monosodium glutamate and pH in order to improve the colorant production. We also made the studies using minority requirements, minority compounds, for example, magnesium to the fermented media, and you saw that you could improve the colorant production. And here is only to show for you the difference of color of the colorant produced with around through the experiments made. But the result here is that monosodium glutamate supports low microorganism growth and high amounts of extracellular colorants. At this point, we are doing together some experiments to extract the colorants in the cell. So we use, for example, solvent organics combined with mechanical disruption cell and with ionic liquid as well, and we have good results. However, this one was more important because during when making a modification in the fermented media, in the media composition, you made that the colorant was extracellular now, and so this extraction process was not more needed during our process. After we began to work with bioreactor, and so we made this scale up, passing for incubator shaker for bioreactor. And the first point that's important to see, when you're going to make experiment bioreactor, generally you have 3 steps. So the first is the pre-inoculum, the second is the inoculum, and the third part is the production. So the experiments in the inoculum, when you have bioreactors at the size of they were working, that was 4 liters, we have this incubator shaker. And we began to analyze the first point, which was the best media for the inoculum way. Because you know that in monosodium glutamate media, we don't have a lot of growth. The microorganism only produce the colorant. Okay, but for put this microorganism inside of the bioreactor, we need amount of cells because the cells first is going to grow and after produce our colorant. So the media with monosodium glutamate for starter culture was not so interesting because the microorganism did not grow and the production of the colorants began there, but we don't have a lot of high amount of colorants produced. And here, when you analyze the microscopy, you see a lot of difference about the kinds of the microorganism. Here, when you are working with monosodium glutamate, you have conidias, conidiophorus, and IFAs, filamentous of IFAs. But when you are working with complex media, using glucose, meat peptone, and meat extract, that is allowing a high growth of the microorganism, what you see a lot of pellets in our media. So this point was very important because here we defined which was the best media for the inoculum step. So what you want? You want not the production of the colorants. We want the microorganism growth, the microorganism in the best metabolic moment for the production of the colorant when you give for them for the monosodium glutamate and the glucose. So we defined it there that the complex media is going to be used as a starter, incubator shaker in the inoculum step. And so we made the first experiments in bioreactor. So we would use a starter tank bioreactor working bat mode at 30 degrees and 100 RPM. And here you change the airflow. So what you saw, when you have, for example, monosodium glutamate glucose or starter culture composition, the colorant production was low and the glucose consumption was also low. But when you use complex medium as starter culture, you had improvement in the colorant production, the glucose was consumed during the process, and you have high amounts, not a high amount, but more biomass than the first one. And the viscosity apparently didn't change a lot The viscosity is generally a problem when you are working with filamentous fungi in sterile tank bioreactor. But when you define the best media and the media with a low growth of the microorganism, it was not a problem for us. So we could use this kind of bioreactor without any problem. And here, analyzing the process in this sterile tank bioreactor, you can see that the microorganism did change a lot about the characteristic here. When you use monosodium glutamate as a starter, the microorganism did not grow a lot, but here it grew a lot. And here you can see that the production of the colorant was very high, and the glucose consumption, it was not so high. As a result of these specific experiments, you can say that the colorant productions by Thalorubicin stokii is dependent on nutritional and environmental conditions, change in nitrogen source, or especially related to the activation of red colorant synthesis because of the biochemistry process. Monosodium glutamate and environmental pH play an important role in the metabolic stress affecting cell growth and colorant productions. The main effect of culture composition was on fungal morphology states and non-directional relation among mycelium accumulation, morphology, and colorant production, and pellet formation favoring suitable oxygenation of the culture growth, leading to the best metabolism of the carbon source. So, it's important to improve the production of the colorants. New study bioreactor is one thing that you have already done, but I'm not present here for you today because it was not published yet. But we worked with different kinds of impilers. Here you are working with Rushton impilers. We are working now with different ones, and with different, we worked with different of oxygen supply. So we work with cascade mode and a regulatory one. So we work with different conditions in the bioreactor. And here you want to optimize production of biometabolic engineering, use systems biology-based models. So what you want now is study more deep the biochemistry of the process. So you know that monosodium glutamate is the best nitrogen source because in a way you have the production directed for the we have the growth, the microorganism metabolism directed for the production of the colorants. However, now we want to know how it works, how we can do this, and how we can improve it. F is better if you give first the glucose and insert the monosodium glutamate in a specific point, or what you can do. Which other amino acids you can insert in order to improve the production of the colorants. Okay, so as final remarks, I can say that there is a large chemical diversity in microorganisms to be exploited, and we have, and if you know, work with high production is very important to make these experiments in incubator shaker and in bioreactor. Incubator shaker, you can define different points. So, for example, media composition, initial pH, time of fermentation, some points. But you have to also work with this transition of incubator shaker for bioreactor, and some points have to be studied, analyzed to improve the production. Other thing is that when you know about the production step, you can improve the downstream process because you can make a strategy to downstream process more applied to your specific process. So you can combine the production and the fermentation process in one single point in order to make the whole bioprocess in the best way. So I'd like to thank you, FAPESP and CNPq by our founding here in our research group, and thank you for your attention. If you had some doubts, I'm going to be glad to answer now, or if you want to send me email after, I'm going to be available to answer it. Thank you so much, and thank you again, Ana Paula and Marianne, for the invitation.
Thank you, Valéria. Thank you very much for your nice presentation. It was very interesting. So now we are open for questions. Any questions?
I have one, if no one else have. Hello, Valeria. Thank you so much for your nice presentation. Well, I have one question that is a more general question. Despite the relevance of these natural colorants, well, I have two questions. Despite the relevance of these natural colorants, do you believe that they can compete with the synthetic ones in terms of economic viability? Do you think that we will reach that place?
We have one specific about monascos that they are going very competitive now, because mainly in Asian countries, as I said, they are working with the production of natural colorants for a long time, and they reduced the production of mycotoxin, and they made the downstream process also to remove the citrulline produced. And I have one script that I made, I read few months ago, that they compared this production. It's not the synthetic one is cheaper, however, the natural ones is cheaper, is more, with the time they're getting cheaper. And so I think that in the next years, they are going to be competitive in the process, mainly because of the toxicity of the synthetic colorants. So if you improve and if you're working with this process and improvement of the process, working with cheaper culture media and working with downstream more efficient, they are going to be competitive, because we have now a very interesting in these colorants. So for example, in Brazil, I am working with the process of production and do have a contact with two industries that are interesting to make this production in a high amount, because they said that they have a specific public that want these natural colorants in the products. And so, although the price is not going to be equal, we have a need in the market for it. And sometimes the market is going to talk more than the price. So I think that in few years, if you're working with this development of the process and improvement of the process, they are going to be competitive.
Yes, I do believe so. At least you will have a fraction of people require natural compounds, and for sure they will pay more. Yeah. But even though, how do you will overcome the problems associated with the usual raised concerns on the use of organic solvents and ionic liquids in the downstream process? Which type of proofs or experiments do you need to do to fulfill the requirements by the regulatory agencies, for instance?
What we're looking now is for organic solvents more green, that are allowed to be used by the regulatory agents. So, for example, ethanol is one of solvents that can be used. And when I said about ionic liquids, is when in the beginning of our research, we have this colorant produced intracellular. We have this extracted in the media, however, had a high amount in the cell. So we would like to remove it. And we began to work with isomeric liquids and organic solvents, and it was good. We could extract it. However, we began to think about the strategy to change the media,
yes,
and not to work long. And we had good results, because now we are producing a high amount of colorant compared with the initial ones, without the problem with the intracellular colorants. But I think that if you think about the strategy, is the best way to work with organic solvent greener, for example, and these strategies that's going to be more acceptable for the regulatory agencies.
Perfect. Thank you so much for your very nice presentation.
Thank you.
Thank you, Valeria.
More questions?
I have one. Well, first of all, it is a pleasure to hear all this work that you have done since your PhD. I already know some of these results,
Yeah.
Well, one of the things is a note for you. So I was talking with Professor Diana, and probably during this next weekend we will have some explanations about the possibility of to characterize completely the structure. So let's cross our fingers.
Oh.
And then the question is, do you know what is changing in the metabolism of the microorganism when you of nitrogen and it starts to produce only extracellular pigments?
It's one thing that I want to begin to study now with biochemistry specific marker, but we have different strategies. One thing is that when you put this, if you remember, you have produced first the orange colorants and you have the amination process, they begin to produce the red, and the red one is the one that's more excreted by the cell. This one is more water soluble. This is one point, and so it can move through the cells. And the other point is that this one we don't know yet because you have to work more specifically with carbon marker and nitrogen marker. That's going to be experiments that I hope we are going to make in the next year. But they change the morphology of the cells because the cells is also change when you change the media, culture media. So depending on the media, whether you're using, they are more thinner or not, and this change also in the process. And there is one thing that we're going to study in the next year to prove our concept. But I don't have sure yet. I have to make some experiments to prove it. But it was good that we could see. I didn't show the pictures, but we saw the difference about the morphology of the cells when you use, for example, monosodium glutamate and you use other complex nitrogen source. They change a lot, and this is about the metabolism of the microorganism, and now is one thing that we are going to work in the next few years.
Okay, thank you, Valéria.
Thank you.
More questions?
I have one question. Hi, Valéria.
Hi, founder.
Very nice talk. In the beginning of your presentation, you said that depending on the fermentation or in shaker or in bioreactor, several conditions affect the fermentation. In your opinion, which conditions affect more the fermentation in shaker and the bioreactor?
One fact is that when you are working with aerobic microorganisms, when you make this in shaker, we cannot study very well when you don't know about the oxygen transference inside the shake flakes. When you pass it for bioreactor, we can work with more. You work now with the oxygenation through the bioreactor. And so this is one point that generally improved a lot the production of biomolecules through aerobic microorganisms. So if you can say that one point that if you work, for example, 100 RPM in the incubator shaker, it's okay. But when you go eat for bioreactor, you have to make a lot of studies about this oxygen transference. And only changing the oxygen transference, we know that we can improve a lot the production. So I think that one specific point is about the oxygen transference in bioreactor and incubator shaker.
Okay. Thank you.
Thank you. More questions?
Yeah. So I have one short question that is about toxicity of, for example, if you use wastes and you do extractions of wastes with some solvents, and then if you use these wastes for fermentation of your Saccharomyces, do you believe that Saccharomyces is able to use raw extracts, or we need to make a high degree of purification of these extracts to ferment the cells?
One result here that you use orange wastes. And when you use directly the orange waste, you don't have the production of the colorants, mainly because the microorganisms produce a lot of enzymes. So we saw that we had a high amount production of chilanase, we had production of cellulase, and the colorants are not produced. So what you were doing now is made the hydrolysis enzymates of these orange wastes in order to have the carbon source available to the microorganism. And we didn't try it yet because we are working with the hydrolysis enzymates point, but we believe that after the hydrolysis process, we're going to use this carbon source. However, when you use directly, the waste is not appropriate because the microorganisms produce a lot of enzymes. And we have this electrophorese, and the electrophorese, we have a high amount of proteins produced together. And so probably the microorganism didn't direct its metabolism for the production of the colorant. Instead, it produced more enzymes. We made some tests working with orange waste and bagasse as well, and we inserted monosodium glutamate. And what we realized is that the monosodium glutamate was not used. So the microorganism, during the time that you worked, you made this experiment for 7 days. One strategy is increase the time of the fermentation. However, increasing the time, you had other kind of problem because it's going to take fermentation like 15 days. It's too long if you're thinking an industry approach. Is not so now what we're doing is making hydrolysis of this waste in order to take this carbon and use the carbon. But we know that when you have bagasse and orange citrus, the microorganisms produce a high amount of enzymes. And the monosodium glutamate is not used. It keeps the same concentration of the beginning.
Yes, I agree with that. It's the best approach is to make the hydrolysis first. If not, we need to wait a long time for the microorganisms to produce the enzymes and then to convert to carbon. And then the colorants. Yeah.
And one strategy was making it longer, but I thought that was not available to work with 15 days of fermentation. Is too long.
Yes, thank you.
More questions? So, no more questions. Thank you again, Valéria, for your very nice presentation. And thank you all to be here today with Valéria and learn a little bit about fermentations and productions. Thank you, Valéria.
Thank you, Ana Paula. It was a pleasure to make this presentation for you.